Dr. Jack Tarlton received his PhD in molecular and cellular biology from the University of South Carolina. He's the retired director of the Fullerton Genetics Center Laboratory of Asheville, North Carolina, and served as director of the Molecular Genetics Lab section. While serving as lab director for over 20 years, he witnessed the explosive growth of genetic information, and as specific genes related to genetic disorders were identified, Dr. Tarlton was drawn to the work of understanding the molecular details of how genes function. Now, I was told at a previous conference that sometimes the blurbs that we're able to get the information about so that I can read them to you may not be all or inclusive. So, Jack, we're very happy to hear from you today and have you here today. And if there's anything that I've neglected to mention, please mention it.
Oh, great. So I don't know if you want to do the podium or the, yeah. Oh, it's up there.
Wow, thank you very much. Is this on? Can you hear me? No? How about now? Okay. Thank you very much. It's an honor to be a participant here, and I came into an interest in periodic paralysis later in life, and I was about ready to retire, and then all of a sudden I have a new job and a fascination with this disorder that affects so many people. And we really want to make sure that when we were talking about genetic testing that we're looking at all the possibilities. And we've had a number of genes that have been isolated over the years, but there are a substantial number of people that don't have one of those mutations. And so I have focused on doing some work to look at patient samples that were collected mainly by Dr. Lehman-Horn over the years. And when his lab closed down, we ended up taking those in our lab and just decided about 2 years ago that we should do a more formal investigation. So what I want to talk about today is some of that work, and it's definitely a work in progress. We're still trying to figure out what would be our goal would be what is the best approach in terms of genetic testing, what's the most cost effective, what's the most technically correct way of doing it, and that's what I'm trying to address today. So let me make sure. Okay, so I picked this slide with a cross-section of muscle tissue just to illustrate how compact everything is tightly packed in. So I'll talk a little bit about the known genes that we know about in terms of the ion channel genes that are located in the sarcolemma, the membrane that surrounds the muscle cells. And these microfibrils are in parallel with each other, and mitochondria will be mentioned today. We'll talk about the powerhouse that makes ATP in the cell and how that might be affecting some individuals. And just kind of the anatomy of the structure of the muscle cell. We'll talk about T-tubules, this network of membrane that goes down inside the cells. So I chose this just to illustrate how tightly packed these literally hundreds of proteins, maybe thousands of proteins are, and how they may affect each other. So just a little bit of background. Our laboratory in Asheville, North Carolina, has done testing for about 25 years, and I got interested in the ion channel maybe about 2005. And when Dr. Lehman-Horn's laboratory started closing down, we ended up doing a lot of testing for the 4 genes that were known, the sodium channel, the calcium channel, and the potassium channel, KCNJ2, and a second calcium channel that's involved in thyrotoxic periodic paralysis, KCNJ18. So that was the standard panel for testing for a long time, and still is to some degree. The KCNJ18 testing it's still performed, but it's not routinely done, I think, much anymore. And that's just because some of the results were a little hard to interpret. So I wanted to talk a little bit about and focus on the disorders that are not the ion channel disorders, those that have the primary disorders, as I define them up here, is that the primary periodic paralyses are those that have a family history, typically, so they're familial. Sometimes they may be brand new mutations, and so they may still fit into the primary category, but they are not necessarily inherited. You can get a new mutation. The secondary causes, these are found in patients that have a disorder that's unrelated to ion channel. So no ion channel variant is present, and these examples of this may be renal disorders and hyperaldosteroidism, and the periodic process that's associated with hyperthyroidism. So those are secondary causes, but what I really want to focus on is the atypical ones. These are patients that have unusual clinical presentations. We do the panel testing, we don't find anything in those genes, and then we're kind of, in the past we've been kind of stuck, but there's been some recent advances that I wanted to talk a little bit about. So examples of these may be the disorders that are associated with the ryanodine receptor, the RYR1 gene, that in recent years we found more and more examples of. And then there's these so-called benign variants in SCN4A, the sodium channel gene, that sometimes get paired. By themselves, they seem like they would be benign when we do our workups, but they seem to be occasionally paired with another variant in a different gene, usually a chloride channel gene, that may actually give rise to symptoms. So, about 2 years ago, we embarked on a somewhat of an informal study to look at some of the families that we had obtained samples on through Dr. Lehman-Horn. And we did a technique that's called whole exome sequencing. So this is a look at not just a single genes, one gene at a time type of thing. We're looking at all 22,000 protein coding genes in the human genome. So part of the problem is that that creates a lot of data, and the analysis of it is really intense and laborious. But it may be that this is the best approach for doing the types of testing that we need to, when we really don't know what gene we're looking for. This is probably the best approach. So we selected some patients and just contacted them, got informed consent for doing the study. So these are patients that are negative for the known pathogenic variants. We used to do that with a technique called Sanger sequencing, which is a 1-by-1 gene. And we used the exome sequencing is what we call next-gen sequencing. So this may be a better approach because we're really on a treasure hunt looking for new alterations and trying to fit new things together. I will say that this is a very limited, it has a lot of limitations to doing this. The real proof of principle is to have functional studies. And so what we're identifying are genes that we might want to focus on, but eventually want to do functional studies on. And that's another level of complexity that we didn't address. So we don't have anything that we can say with absolute precision that we have found a particular gene. I do think we've found several situations that contribute to patients having periodic paralysis without having one of the known primary forms. Just a patient overview is that we contacted about 22 families. We had, most of this work was done in our lab, but we had some work that was done in some of the commercial labs that we analyzed after they were done elsewhere. But typically the best way to do this is to obtain samples from multiple family members, somebody who's affected, and maybe even two people are affected, but usually a parent and a child, or perhaps in some cases we used distantly related cousins that had similar symptoms, similar clinical symptoms, and we wanted to take the measure to find what they had in common with each other from a genetic point of view. All of these patients have a history of partial or full paralytic or severe weakness events that are episodic. Most of them carry a clinical diagnosis of hypokalemia, periodic paralysis. But that sometimes just means that there also could be normokalemic. They tend to respond, most of the patients tended to respond to oral potassium. Most people had the common triggers that you're familiar with. But these patients sometimes seem a little bit more complicated. They may have some other symptoms that I've listed out up here that may actually be contributing to the difficulty in diagnosing them. They're responsive to different medications. Sometimes that's a clue. Diamox would necessarily be effective in all cases. Once we identified what we call a gene variant, a change in the gene. So you'll hear the term mutation and variant used somewhat interchangeably. But typically when we say something's a mutation, we have fairly conclusive evidence that it's a pathogenic alteration. When we identify these variants, these are what I call candidates. We are looking to see, does this make sense? Do they fit physiologically with what we think is going on? So I'll refer to these more as variants than mutations, but sometimes they do cross over and become mutations once we obtain enough proof for deciding that they're really meaningful. So the known mechanisms that we know about to date physiologically is that the sodium and calcium channels that are involved in primary periodic paralysis have what's called a gating current. These alterations, these variants that occur, these mutations that occur in those genes alter the voltage sensing function of the channel, and that's called a gating current. So we know that the mutations cause a little bit of a leak. It's like a little short circuit that occurs there. And in the KCNJ2 gene that's associated with Anderson-Tawil, we know that that's kind of a loss of function of that gene. The gene product is not there in the amount that's a normal amount. It causes its effects related to losing function as opposed to the leaky channels that are kind of a gain of function in the protein. Over the course of the last 6 or 7 years, there have been about half a dozen publications in the medical literature that describe SCN4A variants that seemingly, when we go through all of when we find these, they're not in the typical places that the other sodium channel variants are found. And we go through a lot of analytical process to look and see are these really in a spot in the gene, or are they causing a functional problem that might impair the gene? Typically, by themselves, they seem not to be causing a problem. But occasionally they get paired up independently with an alteration in another gene, and typically in the cases that have been reported, that's been the chloride channel. The chloride channel is an ion channel as well, and the chlorine going in and out of the cell is involved in restoring the resting potential of the muscle membrane and enabling the next cycle for an action potential to come down through. So the gene that's involved in that is the CLCN1, the chloride channel gene, and you may know about that from myotonia congenita, the disorder that's associated with that particular gene. About 2013, there was a study coming from France, from a group there that looked at a number of individuals that had an alteration in the mitochondrial gene that's called ATP6, and another one that's called ATP8. And those two genes overlap. So mitochondria, you may know, are independent organelles inside of the cell, and those are involved in generating ATP. ATP powers a lot of things that go on in the cell. A lot of the chemical reactions wouldn't work without ATP being involved. And the mitochondria in the typical cell, there may be hundreds of those independently circulating there or involved in the cell. And those generate the ATP for powering things, but they do it internally. So there is actually a piece of DNA, a circular piece of DNA inside of every mitochondria, and actually multiple copies of that. And there's 37 genes in the mitochondrial genome, as opposed to the human genome that has 22,000 that's located in the nucleus. So the mitochondrial genes are involved in these bioenergetic pathways, and if ATP production is reduced below a threshold, that actually can impact a lot of the biochemistry that's going on inside the cell. So in 2013, this group found a series of patients that they identified that had these mutations in the mitochondrial genes. And that's as opposed to genes in the nucleus that actually can affect mitochondria as well. And I'll talk a little bit more about that in a moment. About 3 or 4 years ago, the first variants in the RYR1 gene that I mentioned earlier became characterized. That gene has been well studied for a number of years, but these were patients that had episodic weakness, episodic paralytic attacks. They actually had an atypical clinical presentation compared to patients that have primary periodic paralysis. And it turns out that there's, because that's such a big gene, there's probably a fair number of people that had an undiagnosed case that do have RYR1 alterations. And so we've uncovered in our series of patients something like 30% of those patients have an RYR1 variant that we are suspicious of. We can't definitively say, but we think it probably is related. And then about a year ago, there was a publication about this time last year looking at the sodium-potassium pumps. These are proteins that are embedded in the muscle membrane that actually shuttle potassium and sodium in and out of the cell. And so they're cyclical, so they're pumping it in and pumping it out. So there's a gene called ATP1A2 that was reported, a patient was reported to have. It's a study that came from a group in Brazil and collaborating with a group in London that Dr. Hanna is involved in. And they identified one patient so far that has this alteration, only one that I'm aware of. So those are the mechanisms that are known already. I borrowed this from this illustration from Dr. Cannon's, one of his papers from a couple of years ago, a commentary that he did. And I just wanted to point out, if you look on the left-hand side, that's the sodium channel. That's a cartoon of the sodium channel as it winds its way. So that ribbon is a string of amino acids, which is at the genetic level, that's the gene encodes the precise order of those amino acids, and they have certain biochemical properties. And the one on the left side of that slide is the sodium channel. The right is the calcium channel, two independent genes. But you can see how similar those channels are, and you can the notation there is looking at the voltage sensing portion of the gene, of the channel, and mutations that are in that. So we have a very good understanding, thanks to people like Dr. Cannon, for understanding the sodium channel and those leaky currents that occur there that give rise to primary periodic paralysis. And I wanted to say that I put this up just because that's well understood. The mitochondrial, the sodium-potassium pumps, the RYR1 variants involved in the ryanodine receptor gene, those are not very well understood. So we're kind of at the cusp now of moving into a greater understanding of kind of the broader spectrum of genetic disorders that give rise to periodic paralysis. So this is the paper I was mentioning from the group in France, and so the title there is in orange. So it's episodic weakness due to mitochondrial DNA mutations in the ATP6 and 8 gene. There's a lot of technical things that go on related to mitochondrial function that really haven't been well characterized. But these deleterious mutations occur in a substantial number, if not close to 100% of the chromosomes that are the mitochondrial chromosomes. And these patients had episodic weaknesses, but they were actually diamox. Acetylcholine was actually very effective in treating these patients. And so there's only this report of the mitochondrial disorders that have been confirmed. I have yet to see a second follow-up on this. So this may be a rare cause, but I think it's something, the energetics that are involved in mitochondrial function are really powering a lot of things. So even the sodium-potassium pumps, they take a lot of ATP to actually work. So things that fall below some bioenergetic threshold may be impacted by a reduction in function, if not a complete loss of function in the mitochondria. So the paper about 2 or 3 years ago that came out related to RYR1. This was actually the second report of RYR1 having involvement in periodic paralysis. But the original paper, I think, came out in 2010. But the paper reported on 3 additional patients that had alterations, had variants in RYR1. So the cartoon that I've put there is just pointing out. So the calcium channel, so this is one of those T-tubules that I was mentioning earlier. So the calcium channel is here and goes through the sarcolemma membrane, but it actually is in contact with the ryanodine receptor. And this is the sarcoplasmic reticulum that is loaded with calcium, and calcium is really the driving ion for muscle contraction. So you can understand why if something that comes in contact with the calcium channel has some alteration in it, it may give you a clinical manifestation of something that's similar to the calcium channel phenotype that comes with mutations in that particular gene. So that makes sense that there would be some connection there with the ryanodine, and maybe there are other genes, products here that may be involved in the future. If we, as we study this a little bit more, we may find some of these other alterations there. So this time last year, this ATP1A2 was reported. So this is, as I mentioned, was from a Brazilian group in collaboration with the group and with Dr. Hanna's group in London. And so they found that the variant that was occurring in this particular protein was really related to having a leaky mechanism in a similar fashion, analogous fashion to the chloride channel, or the sodium channel and calcium channel. This patient actually had seizures as well, so an atypical clinical presentation. The protein that comes from the ATP1A2 gene actually is tissue-specific in muscle, is expressed in muscle, and it's also expressed in neuronal tissue. So the seizures kind of make sense that are a part of that spectrum as well. So most of this adds up in a logical way. I put a lot of verbiage here in this slide because I wanted this, when it goes into the meeting notes, for there to be some context. But these are places along the spectrum of the action, muscle action potential that potentially could lead to, if there are defects in proteins in particular areas, these may lead to clinical manifestations. So at the neuromuscular junction where the motor neuron interacts with the muscle fibers, there's about 50 proteins or so that are involved in making a normally functioning neuromuscular junction. And so there may be places, and we know that myasthenic types of symptoms originate from some of the defects in these particular proteins. And that might be a place when we're looking for alternative atypical types of genetic alterations that may be giving rise to periodic paralysis. We should look closely at the neuromuscular junction. And I mentioned to you the primary forms that are coming from the chloride channel and the calcium channel and the sodium channel. And I mentioned that kind of in the second here. These muscle action potentials are dependent on those working in concert with each other. The calcium that's released during after the muscle potential interacts with proteins, structural proteins that are troponin and tropomyosin that are in the structural parts of those myofibrils. And there are no known causes for periodic paralysis related to those. And I'm speculating that there may be some possibility of that, and I want to tell you a little bit more about that in some of these other slides. So we did this whole exome sequencing for a number of patients, about 22 families total. Sometimes we only sequenced one person. Sometimes we sequenced two affected people just to see if they would match up in some fashion or form. And these are variants that were found there, but I just wanted to pick a couple out and just mention them to you. So you can see at the very first one, there's an RYR1 variant. There are a number of RYR1 variants scattered through here, as you can see. And I mentioned earlier that we had a pretty high percentage of those compared to other alterations. We found some patients that had nothing that we could identify as a primary cause, but had a cluster of variants that would be associated with those proteins in the neuromuscular junction. So that became kind of a suspicious avenue for us to pursue. And we looked somewhat closely at those. Here's another part of those the series, and we found any number of variants that we then had to go and explore one by one. And these, just to summarize the study findings, is that we found 4 patients that had sodium channel SCN4A alterations that by themselves we would have judged to be probably non-causal in terms of causing the independently causing symptoms. And in some cases these were paired up with the chloride channel variants that I mentioned to you earlier. And because that had been reported before, I think we were able to come up with a somewhat of a nice story for those families. And at least we've given a pathway that we can kind of go down. We had 6 patients out of the 22 that had RYR1 variants that we are suspicious could be involved in the pathophysiology. We would call these as having we describe them as having uncertain significance because we have yet to do the functional studies on them. So that would be the gold standard for establishing them. We didn't find any patients that had alterations in sodium potassium pumps. And then there were 4 patients that we did not have any strong candidates for. But we did have several patients that did have these unusual alterations, and I wanted to really label these as speculative. These are not definitive at all, but they're suspicious in terms of having alterations cluster in a particular place. So we had 2 patients that had seemingly had a number of different alterations that potentially could be involved in disrupting the neuromuscular junction. And interestingly, 2 of these patients really responded well to medications that are known to cause the acetylcholine to linger and it reinforces the medications actually are treatments for the myasthenic disorders. So these patients, although they had episodic paralysis and weaknesses, seem maybe to have something more along the myasthenia part of the spectrum. And these patients had dramatic improvements when they took mestinone and 3,4-DAP, the diaminopyridine, and it really reinforced our thinking that maybe that's something going on related to the neuromuscular junction. But again, these are speculative. We really have not established that at all. And the other thing that we found was a couple of patients that had mutations in filamin C, which is one of the structural proteins that's involved in the myofibril function. And these patients probably don't have periodic paralysis, but they do have severe attacks of weakness, and it may be mimicking periodic paralysis. And they have a pretty complex clinical presentation that includes some cardiomyopathy. And it made us think a little bit about these myofibril types of structural proteins. The myofibrillar myopathies are a group of disorders related to defects in those genes. And sometimes it may be that some of our patients that are showing symptoms that look similar to periodic paralysis may actually have something going on in those. And we did have one patient that had a caveolin 3 mutation that is a known pathogenic alteration. So that's a variant that didn't need particular workup. But he also had a defect in another gene called POLG, polymerase gamma. This is a gene that's involved in normal functioning for mitochondria. The mitochondria would, if it's being affected, might be contributing to the pathology that goes along with the caveolin 3 defect that we found. So this patient actually had some episodes that looked very similar to periodic paralysis and was wondering whether that's a true case or not. We found other patients that seem to have defects that would be affecting the nuclear encoded mitochondrial genes that affect mitochondrial function. And so those are candidates in terms of affecting or interacting with these other genes. So we did find one family that has an SCN4A sodium channel variant that we would typically label as being benign, but also had a thymidine phosphorylase deficiency or an alteration that we speculate might lead to a deficiency of this enzyme. And that, the TYMP gene is a gene when both copies of it, one that you inherit from mom, one that you inherit from dad, are affected. That gene can actually give rise to a mitochondrial disease that's involved in depletion of mitochondria. So again, these are all speculative. We don't have real evidence, but it's giving us some pathways to pursue, and that's really what we want to do in the upcoming years. And I think I've kind of mentioned these associations here. So in summary, these atypical periodic paralysis associations often come with a complex of symptoms that really don't fit nicely into the established primary forms. So we've got kind of the defining hallmark characteristic is these episodic events that we all are familiar with, but they may have other things going on that really contributes to a more complex presentation. The RYR1 disorders, I think, are really going to be established as a maybe an atypical form, but to go hand in hand with the primary forms in terms of what we do first when we do testing. And of course, the testing that's being offered through Strongbridge includes the RYR1 gene. And so just to give you an illustration of how complex that gene is, it's about 4 times larger than any of the other ones. So when we start to do the work of analyzing genes, we have to go through a lot more data when we look at RYR1. We really couldn't possibly have done that testing with the older techniques. It's such a large gene. It just is not cost effective to do. It would take months to get a single gene done. So the exome sequencing or targeted genomic sequencing, where we do, target maybe 200 or 500 genes in a single testing, would probably be the best way to go. So the genomic approach, I think, is supplanting the old 1-by-1 gene. One thing that we don't have a definitive handle on is what other genes for these disorders that seem like they may be, that they're not the primary forms or not the ones that we have a suspicion about at this point. Should we be adding a number of other genes to our panel to be looking and screening some of the other neuromuscular disorders that may be related? We don't have the data to support doing that just haphazardly, but I think most of the testing that's being done by Invitae and some of the other commercial companies will, they also have an option for including other neuromuscular disorders, and maybe something like the filament C that I mentioned earlier, these myofibrillar myopathies that may actually contribute to some misdiagnosis may be covered by those. We also need to have better recommendations for how we do the variant interpretation. So there's a lot of complexity there, and it would be nice if we had a working group that actually standardized that. We're getting a lot of commercial lab derived sequences that are available to patients, and patients really don't have an idea of how to analyze those, so they come looking for help, and there are not many of us around that have experience in looking at periodic paralysis genes. And so it would be nice if we had a standard way of approaching that in terms of taking data that's produced in a number of different settings, either in clinical diagnostic labs or in laboratories like Dante, which a lot of people have had their genome sequenced. It's great to get your genome sequenced for, a few $100s, but it's the data interpretation that really is the critical part, and that's much more difficult to do. And in clinical labs that do genomic testing, that testing typically would cost several $1,000s to actually get the analytical part done. And it would be nice if we had a targeted group that we could focus on to do that. And then kind of lastly, it would be interesting if we could take some of our cases that are atypical ones that we don't have an explanation for, and have group discussions of those in case conferences. And I think that would be a noble goal to do in terms of something that maybe the PPA could help us organize. So I'm going to stop there, and I'll be glad to take any questions. I'm not quite sure how much time I have. How close are we on time? Okay. Just move on.
Oh, yeah, so if there's any questions. Yeah. Okay, if there are any questions from the audience.
Yes. So, okay.
I think you kind of touched on this a little bit towards the end, but I'm one of those atypical people. Sorry. I think you kind of touched on this a little bit at the end, but I'm one of the atypical people where they have not found a gene, but they've clinically diagnosed me with this. And I would really like to be able to volunteer myself for the research community, but I don't have any way to do that. So, I mean, I didn't know if there is anything set up anywhere where I can say, hey, take my DNA, hey, ask me these questions, you know, and if there's anything set up like that anywhere where I can volunteer my information.
So the question is there a mechanism for being involved in the research side? And at this point, there's not. We really have, it would be nice if we had another laboratory that's actually doing some of the work as well. Our laboratory is in a hospital that for 125 years was a nonprofit. And then about 9 months ago, we got bought up by Hospital Corporation of America and became part of corporate America. And their bottom line does not include doing a lot of those studies. So we're kind of left with piecing things together in terms of that effort. So there's not really a concerted effort at this point. I fully agree that it would be something that we should try to, maybe the PPA could actually take some initiative in that regard in helping organize that. We desperately need others that would be interested in doing the genomic types of sequence, looking at the atypical cases. And there's plenty of opportunity there for young researchers that are interested in making their mark. These would be golden opportunities, I think. But at this point, we really have limited possibilities for volunteering at this point.
Oh my gosh.
Okay.
What? No, there's more. Just
Got one more. Got one more back here.
Hi, I was at the 2008 conference, and both my children and my granddaughter were genetically tested by Dr. Lehman Horn. They have hyperkalemic periodic paralysis, but what he was telling me is he felt like they had a variant on it, and I was wondering, since you were collecting that you did have some of the blood from Dr. Lehman's Horn testing, would it be possible?
I am not hearing much.
Oh.
Could you?
So she was saying that do you hear this? That her blood was with Frank, and there was a question coming.
Both my children and my granddaughter were genetically tested with hyperkalemic periodic paralysis by Dr. Lehman Horn.
Right.
And at the time I was talking to him, this was in 2008, I was talking to him about the different symptoms, and he felt like there was another variant, another defect on their gene. And I was wondering, since you had the blood and was from Dr. Lehman Horn, could you do more testing to see if there is something additional other than the hyperkalemic?
Right. I think those samples that were collected in Dr. Lehman Horn's lab in Ulm are available for study if somebody really wanted something done that's different than what we've done already. We have not studied, we've not gone back and looked at each one of those, and there's probably something like close to 100 of those. So there's definitely some capacity for doing additional work. What we really would need would be just a personal contact to let us know that you have had some work done, and then there may be some follow-up that we may need to do. So if you would just contact me individually, that we could maybe have a conversation about that.
One more here.
So my mom and I both have the benign SCN4A and long QT, and our triggers are mainly salt and sugar. So our doctors are pretty sure there's one other genetical defect involved, and they're trying to find it, but I'm not sure where to look. Can you might give some hints? I don't know. Do you know anything?
Jake, I'm not hearing good. Can you interpret that for me?
So there are SCN4A variants, and I'm sorry, just repeat.
So we have the benign SCN4A, we have long QT, and our main triggers are salt and sugar.
Okay, so long QT, salt, sugar triggers, SCN4A. Is there another, their physicians suspect there's a second hit. Do you have a sense of where to look?
Typically, the chloride channel would be the first place to look. And, you know, just based on one or two patients that we've done in this study, we should probably look at some of these mitochondrial depletion disorders in terms of being a second hit that might be aggravating the SCN4A.
I think just maybe in the interest of time we move on. I'm not sure. We could do that. The global thing here is, you know, you have symptoms, and then you say, does it matter what genetic test you have or not if you take therapy and you respond to therapy? But then you want to know what you have, and if you have a positive genetic test, does it imply that you should do therapy A or therapy B? And it's still kind of a trial and error, and it's kind of like, what comes first, the chicken or the egg? I mean, is that? So we're kind of stuck in a little bit of a. At least we know for sure there are some definite genes that's our starting point. But now Jack is introducing ideas that, well, there's a whole lot of other proteins that we could look at, that if we have enough folks that are without the routine genes, you look at these other genes, and if the symptoms and those genes match up, at some point you say, you know what, that is definitely real and not just random. But it's hard to get a definitive answer, up front. I mean, is that fair?
I think with these atypical disorders that we're kind of blazing some new trails in terms of trying to understand them. There are any number of genes that we could actually think about adding to the targeted list, and just what I would like to see would be some standardization in the thinking process about that.
So I'll just make a comment. This is Steve Cannon. So it's a two-step process. One, for the atypical cases, it's complex genetics, as you've pointed out here. And when you're talking about novel genes that have not previously been firmly associated with periodic paralysis, that's an extraordinarily difficult nut to crack. And you're right about the approaches. I should let everyone else know who's frustrated that they've gotten back reports with variants in conventional genes like SCN4A or CNA1S, that in many cases you can make an informed approximation and guess what the implication of one of these point mutations will have. But for one of us to study it in the lab, it takes one person 3 months at 100% effort to test one mutation. So you're not going to have high throughput functional testing of ion channels. It's just a difficult problem.
That's a great illustration of the difficulty that's involved in kind of bridging the gap between an observation and a genetic variant and getting to a agreement that is actually pathological. And that's the dilemma that we have right now.
Now, there was a Nobel Prize awarded recently for oxygen and how it responds in different environments within the cell. Is there any part of that research that might help with, how calcium and chloride and other things that are affecting us Is there anything within that research that you've seen that might help with this? They're pointing at each other.
Again, I don't want to monopolize Jack's time, but an interesting development in the field is thinking about the consequences of having these conventional periodic paralysis associated mutations sitting there in the background all the time, but what brings out the episodic attacks? And I know the focus has historically been on potassium, which is appropriate, but we're learning it's much richer than that. I'll talk a little bit about this tomorrow. So it's not surprising that there might be many other systems, not only oxidative stress and oxygen sensing and things like that, because muscle is an amazingly adaptive tissue because the demands, the metabolic demands and the power changes radically. And so I'm sure some of these things will be tied in, but it's complicated.
I think what we're going to do now, if possible, we'll take a break for like 10 minutes. Bio break, bathrooms are over there. Chat amongst yourselves, and we'll be back in like 10, 15 minutes.